Hello, everyone here, and also hello, everyone online. It is always a great pleasure to be here at the NanoN user meeting. And today I am really, really honored to talk about peptide differentiation and identification with nanopores in real time. So when it comes to nanopores and nanopore measurements, the first thing, which is really a key thing to keep in mind when we talk about these measurements, is that you can run these nanopores in two regimes. So the first, which is the more, I think, prominent regime, is called the threading regime, and this is actually employed by Oxford Nanopore for DNA sequencing. And in this regime, you have a motor enzyme which is running on a DNA strand, actually for DNA sequencing, and this enzyme is then used to control the translocation speed of the polymer through the pore, which then is reading out the sequence of whatever is in the reading spot of the pore. And there was also actually a bunch of publications in 2021 where they show that when they chemically link a peptide to a DNA strand, they can use these motor enzymes in order to pull through the peptide chain and read out the peptide sequence in there with the nanopore. So as already mentioned, there's another approach. So how to use nanopores, and this is not showing up. Now here it is. And this is the regime which we call the trapping regime, and this is what I use in my experiments and what I will talk about today. And in this regime, actually, entire molecules are entering the pore. So each atom of a single molecule is contributing to the signal we are measuring, and we call these signals resistive pulses. So you can already see here by the sketched out traces that the signals look a bit different, and this is simply due to the fact that here in the threading we look at subunits, while with the trapping we look really at entities of molecules. And today's topic, I hope, is that I can convince you how we can really recognize sequences of peptides while they are trapped in such a nanopore. And just to give you an idea of how we do that, it's not advancing. Okay. So if you think of a random peptide sequence like this and how you would really read out the sequence of this peptide, the first thing which might come to your mind is that you simply chop off amino acid after amino acid from one terminus, and then you look at what you have cleaved from the peptide, right? But if you do this, you will immediately end up facing two limitations of your experiment. The first being, I mean, these experiments are done in solution, so this is all a free solution approach now. So you would require a single amino acid sensitivity in order to detect them in the pore, and this is something which is currently not achieved due to bandwidth limitation. So maybe this can be done in a few years. But however, there's an even more severe limitation, being that you need also to make sure that the cleaved amino acids will go and into your nanopore and enter the reading zone in the native sequence, because otherwise you will get a misinterpretation of the peptide sequence. So the idea we had actually is we have something else in our solution when we do this constant truncation of the peptides, which is something we call a peptide ladder. And we had the idea that we might be able to get actually some sequence information about the peptide from such a peptide ladder. And if so, we would thereby be able to become capture sequence independent, because we have all these peptides of the ladder present at the same time in our solution. And then this is actually a parallel detection of all these peptides, which is then very fast. So the experimental approach we had was actually to generate the ladder here and throw it on a nanopore. In my case, the workhorse is Aerolysin, and then simply collect some resistive pulses. So these here are raw current recordings here we did. Then from these resistive pulses, we can compute some very nice histograms. And here comes now the trick, because so this is actually a sketched out one. The actual ones I will show them in the next slide. So here comes the trick of this method. So when I hope you agree with me that each of these peptides will maybe produce a unique amplitude due to its size. And then if we analyze here two adjacent peaks to each other, the difference between them is then somehow caused by the amino acid which was lost. So this is then more or less an indirect way of getting an idea what was there actually in the peptide sequence. So as said, we did these experiments. So we synthesized some peptides. So this ladder for this decapeptide here, and I started to add them to the Aerolysin nanopore one after another, just to make sure we will not misinterpret some of the pulses we see. And as you can see, whenever I added another peptide, there is another peak coming up in our dwell-time scatter plot. And in the end, for the 7 peptides of this ladder, we found also 7 signals here, which was already a quite cool thing. But the even more funny thing here, what you can see with your bare eyes, is that the peaks indeed are not equidistantly, so they have a different distance to each other. So for instance, here, the 2 peaks on the very left, which correspond to a small serine that is cleaved, they're closer to each other than the next one, which have a big gap, which might be the bulky arginine that is lost here. But okay, this is a very cool result. So this already goes in the right direction. So maybe we can push this approach a little bit. And what I did then is that I took this decapeptide sequence, and I scrambled it. So I changed it in the sequence, but I kept all the amino acids in there. So they are isobaric in the end. And I did not do this completely random. So we implemented some features here, being some cassette translations like this SRAS motif is then shifted then always by 1 amino acid. We made some conversions, but we of course also kept identity of some residues inside these peptides. And to make a long story short, in the end, we were able to identify all these structural features which we had implemented in our peptide pool. And this, in the end, allowed us in a double-blind study together with Katja from Ionera that we could identify basically from this 50 peptide ladder pool already 2 in a double-blind thing based on a fingerprinting assay. So the next thing is, I mean, the idea or the overall goal of this approach would then be de novo sequencing of peptides, right? And so for this, we need something like a calibration method, because we really would like to have to see, okay, this peak here is shifted by a certain value, and this corresponds to an amino acid, which is, I don't know, serine. And to figure out if we can really calibrate these things, I plotted here, this is maybe a bit of an unusual plot. So what you see here is the delta added resistance. So this is more or less a measure for the contribution of the cleaved amino acid or the peptide actually to the overall blockade, and plotted against the peptide volume. And if you then start to group whatever of your, so all the cleaving products you have here, you group them according to the identity of the amino acids. I found something like this, which already shows, okay, we have here the red dots correspond to arginines. They are all maybe somewhere down here. Small serines are all up there, and the alanines are here. And when we then did some homoarginine ladder measurement, this is the red line which is there, it really felt in the same area than all the hetero peptide events did. So this was something which I found very nice. But then actually Priyanka, another PhD student in Jan Berend's group, she started then to measure a homolysin ladder. And homolysin did a very funny thing. So it started to curve here. And then she went on and did the tyrosine. So this is another funky guy. So this goes exactly in the opposite direction. And the alanine is pretty good according to the heteropeptides, but serine again is doing some crazy stuff, and we are not really understanding this at the moment, but this is the part of our current work, really to understand what is causing these deviations from the expected values. And the first idea we have here is that actually the neighbors in these peptides are really important. So the neighboring amino acids to the terminal ones. So if you look here, in a zoom out in more detail, and we look, for instance, here at the arginine cleaving products. So the red line again is the homoarginine, and whenever we have a tyrosine or a serine next to the ultimate arginine which we are cleaving, so the penultimate position, we get a minor contribution of these peptides or of the amino acids to the overall signal of the peptide. Whereas when we have an alanine here, they're almost identical to the homoarginine ladder. And the idea we had here is basically that amino acids like serine or tyrosine, they may actually undergo some intramolecular interaction by dislocating the charge density of the arginines, and thereby forming substructures of the peptides, which in the end will make the peptides to appear a bit smaller. Whereas when there is an alanine, there is no interaction and no charge dislocation. So This is more close to the situation of the homoarginine peptide, where the charged residues, all of them are positively charged, so they don't like each other. So it is the idea that they maybe form the largest volume in this system. So far, all the results I've shown you are generated from synthesized peptides. So all the letters were synthesized peptides. But also, this is another thing which we have to take care of, is that in an ultimately really de novo sequencing approach, we need to be able to generate such a peptide letter in situ in such a way. And this is an experiment I did together with my master student Jessica. And what she did here is actually she started only with this decapeptide, and you measured for a few minutes the resistive pulses, and what you can see here is the histogram indicating mainly this decapeptide. But if you zoom in very detailed, you will find free impurities from the synthesis of this peptide. But then she added some exopeptidase, and after a few minutes or seconds, actually, she was already able to see here smaller species coming up. And if you compute an histogram here for this yellow area, and you compare it with the histograms which we have collected from our synthesized peptides, we found pretty good agreement between all the peaks which we are looking for. So actually, this means in the end that these exopeptidases are indeed active in a solution. I mean, I forgot to mention this in the beginning. We are working here in 4 molar KCl. Yeah, this is far off from physiological conditions, and this is actually very surprising. And they produce exactly what we want them to do. And this is not all. So what we can also get insights from these measurements is actually we can measure the kinetics of these enzymes. So these aminopeptidase 1, it's supposed to be a quite substrate unspecific peptidase. So whatever it finds, it is supposed just to chop off the ultimate amino acid. But what we found, if we trace the appearance of individual peptides over time, is that we can make these time histograms, and we can really see that the time courses are very different for different peptides. So this means these aminopeptidases are actually more substrate specific than people thought. So with this, I would like already to conclude the first half of my talk. And I hope that you agree with me that the peptide letters can really indeed be generated in an enzymatic approach in a very challenging electrolytes, and that we can indeed use these letter things to deduce some sequence information. And so this, in the end, gives us great hope that this is a really highly parallelizable, label-free, low-cost method for peptide sequence recognition. And especially, this is something I did not talk today about. This method is capable of discriminating positional isomers of PTMs and also of the amino acids, which we have shown already in some other publications here. So for the second half of my talk, this is actually, Gregor gave a very good introduction to that, because we are also interested in other pores and how they behave. And the question actually is that, so I took on a story of a new pore-like protein, which we thought might be also an aerolysin-like thing. And I will show you how we actually started to investigate this protein. So here you see on the left side, this is the aerolysin pore from side and top, and on the very left, this is the new guy in the field. This is called sorrel. So this is a lectin-like pore, so it has some lectin behavior. It's from a marine eukaryote, and when you do a sequence alignment of aerolysin and sorrel, you can see here in blue which part of the sequence are conserved. So there is actually a lot of similarity between those pores in the sequence. The main being the beta barrel, which is almost identical, but also the C terminus, and in something which is called the receptor binding site. So aerolysin has also a receptor binding site, but there's a big debate going on in literature since almost 20 years. So what is this receptor binding site really doing? And until today, this is not very clear. So that, okay, maybe we can simply start, and as we always do, we use the orbits, and we started to try to reconstitute the sorrel pores on the orbit system in our standard DPHPC membranes. But it did not matter how hard we tried. So we could apply like 100,000 times the concentration which we usually use for aerolysin. We did not see insertion of this sorrel pore. Then we tried to play around with the lipid composition. So what we started then, we came up with this DOPC cholesterol GB3. GB3, this is the ceramide, which is then recognized by the receptor binding site of sorrel. But we could also not get any reconstruction of the pore in these membranes here. And this was surprising because it was shown that for vesicles, this works, this composition. But if you do such a membrane out of an octane solution, we cannot get this. So the obvious idea here was actually, is this receptor really present in the membrane? In order to prove that, the idea was to go to the Meca-Opto. Those of you who don't know what the Meca-Opto system is, this is basically the standard or a very similar setup to the regular Meca system of anion, where you have a trans compartment which is then optically accessible. So you could use confocal microscopy. And the idea was actually to use another protein which is called leg A and label this because we know very well that this binds GB3 very efficiently in order to illuminate then the GB3 which is present in the membrane. And here on the left side you can see a Z scan through the Meca-Opto. So here's the photoresist which forms the cavity, the glass bottom, here's the ring electrode of this optically accessible thing. And what you see is basically nothing. And when we did a positive control, so we added a unspecific membrane dye. This is a different story because this was actually what we were also expecting here. So what you can see here, this is the stained membrane. So we had the membrane there. The protein is just in solution, but it's not agglomerating at the membrane, which in the end led the conclusion that we don't have GB3 present in this membrane. And this is actually not very surprising because when you look in the data sheets of these GB3 molecules, they're only dissolvable in chloroform and methanol, which will actually destabilize your bilayers. So we cannot use that. And whatever solvent out there, it will not dissolve this thing. So only lipid domains, but in the octane, where we have still the solvent present in the membrane, it will most probably just precipitate. And then the idea was, because we still were very eager to see whether this sorrel is able to form pores, we had the idea of doing a patch clamp assay in which we use whole cells of a cell line, this H1299, where we know that they overexpress GB3 on the cell surface, and then simply go to the patch liner, put it on the patch liner, add the sorrel, and then monitor the current over time, whether we would see then the gigaseal to go down. And this is actually what we did here. And what you can see here, this is this beautiful heat plot. And I like to show this heat plot because it actually tells you how clear the result is we had. And I try to guide you a bit through here. So the key thing is these upper four lines from left to right, they are GB3 positive cells. Here we had GB3 negative cells. So we treated them with PPMP, which is an agent which suppresses the expression of the GB3. And below there, the other two lines is the very same thing, just we harvested the cells in a different way. And then here, the first 8 columns here, this is the sorrel-1 insertion, and you can clearly see that whenever we have a GB3 in the membrane, sorrel is really inserting, and the gray color here tells you basically that the resistance, which we had in a seal, went down. So we really saw some ionic change over the membrane due to the sorrel insertion. Whereas when we did not have the GB3 present in the membrane, we could not find such an insertion. However, Aerolysin, which you can see with those 4 columns here, it inserts in whatever cell we give it. So it does not care about this GB3 receptor, and it's very fast. So Aerolysin, it was in a similar concentration rate, but it inserted like in a tenfold faster than the sorrel, so which already points into the direction that we might actually be able to measure some insertion kinetics with the synchro patch. And also here, this shigatoxin B, this is another lectin, which is supposed to form pore-like structures on the membrane or actually damage the membrane at a very slow time scale. You can see here, this is this blue color. So it happens, and if you look very closely, maybe not from outside, maybe the people online can watch that. Also, the resistance goes down over time, but with a very low time constant. And when we saw this, we were really very happy on here and said, okay, this is maybe we can quantify this result a little bit. And this is then what Tom helped us here a lot with. So what you can see here is actually a nice dose and response curve, which we could obtain from the sorrel insertion. And again, if we have a GB3 negative, nothing inserts. Another control experiment we did is we used PMPG for a competitive hindrance of the sarol. So it is a soluble ceramide. And again, we could not find any insertion here. So this is a way to control your insertion of pore maybe. And we measured some IV curves from this thing. So we found that it is almost not rectifying. So good behavior, but it is shifted overall slightly into negative potential. So maybe we have a slight selectivity here in this channel, but this is something we really need to verify in further experiments. We also tried different cell lines. So in comparison to H1299, we also tried erythrocytes. And what we found here is that the dose response curve shows clearly that the insertion in these erythrocytes is way less than that. And I'm actually quite happy about this because those were mine erythrocytes. And this H1299 is a cancer cell. So thank God. And in order to make really sure that we have your pores and not just a loss of the leak here, we started to block the pore. And what we did here is we added polyethylene glycol. And at the very moment when we added the polyethylene glycol, the overall current went down, so the resistance went up, which indicates a blocking of the pore, whereas when we, in the control plates or the control part of the same chip, we did not see any of these things, so the current was stable here. So this was really a pack-induced blocking. And yeah, with this, I hope that you agree with me that sarol really forms the pores, but just not in our lipid bilayer system. So it needs the native environment of a cell. And so both aerolysin and sarol have apparently a receptor binding site, but for the sarol pore, this is very, very more selective and very more specific than it is for aerolysin. And this is a very interesting thing, which in the end made or led to the conclusion that maybe we can get some honey from this, right? So we could use here the SynchroPatch platform as a tool in order to characterize these insertions of nanopores into several cell lines. And maybe if we do some molecular engineering, we can understand which part of these pores is actually corresponding or responsible for the selectivity of the pore. And with this, I would like to thank you for your attention and also my supervisor, Professor Behrens, my student, Jessica, as well as Tom and Marcus from Nanayan for the great support here with these SynchroPatch experiments, and also for Katja from Ionera, who helped with this double-blind study in the peptide recognition topic. Thank you.
Thank you very much, Tobias, for this talk. For me, it's quite mind-blowing which strategies you can apply to hunt down such a pore if it's not that easy to observe under standard conditions.
Thank God to the ballpark of Nanayan.
Yes. Are there any questions about this? Yes.
Yeah, thanks. Really impressive stuff that you present as always. Very nice. And of course, I like to see, not only the bilayer system being used for SynchroPatch, really nice. My question regards to your letter peptide sequencing. You showed this 7 amino acid length peptide. What is sort of the limit? How long can you possibly make that? Because I would expect that if you get a little longer, the differences in size that you can possibly detect by blockage or different levels of blockage become very tiny pretty soon. How long can it possibly be?
Thank you, Niels. This is actually a very good question, and we are already tackling that down. And what we can see is we have with the decapeptide, we are almost at the sweet spot of aerolysin. So we can squeeze a bit longer into the pore, so like 15 amino acids is also possible. So we can get a signal, but we are then less sensitive. So we cannot really measure those very, very tiny differences or even isomers. Then it's really just, okay, we cleave one amino acid, okay, this is something detectable, but this very miniature shifts in our current blockades, this is then not detectable anymore. So for this, we could come up with an idea of elongate the channel, for instance, and engineer it. And on the other side, actually, we have a limit currently. This is why it's indicated here with this dashed line, R3. So a tri-arginine peptide. This is the smallest peptide we can currently measure. So this is most of the time it's bandwidth limitation because when they are very small, they just run through the pore very, very quickly. But this is again molecular engineering of the pores, which is key to that. But,
Great talk, Tobias. Very, very nice. How specific do you think the pore forming is in cancer cell lines? Do you think it's, I mean, is it a potential therapeutic, you think, or?
yeah, this is what people think actually, that this GB3 is a glycoprolipid, so a ceramide, which is somehow related to cancer, and most cancer cells actually express it or overexpress it on the surface. So it seems to be quite specific. But as seen with my erythrocytes, there's also something which is interacting with the same site. So
I'm wondering, like, if you use sort of HEK cells or something,
not yet. Not yet.
non-cancer cell line, whether you would see the same thing or.
That's a very good idea. So we need to do that. So what I've shown here with the SynchroPatch study, this was just the very first shot of experiments. So it's very preliminary, but in the end, indeed, this is what we need to do the next time. So now compare different cell lines and see whether we can find differences there. Yeah.
More work for Tom or SynchroPatch in Freiburg then, or?
That would be cool.